What's the difference between a small classroom and a giant university lecture hall? Well, the answer is in the question. One of them is much larger than the other. But if we start to look at similarities, we know that both of them will have a teacher or professor in charge and students attending the lecture. So what's the similarity then between a simple carbon compound like carbon dioxide and something like a benzene ring? Well, there are some obvious differences, of course. Oxygen as opposed to hydrogen, double bonds only versus some single bonds, some double bonds, and obviously the presence of 6 carbons. But the main similarity here is that we refer to both of these as organic molecules or organic compounds. The reason is this one element, carbon. Any molecule or compound containing carbon is an organic molecule. And because of that, they have very unique properties. Now, there's an entire branch for this called organic chemistry, but it's way too extensive for us to get into right now. So, all we need to know for now is that in our situation, the teacher is like carbon. It's what differentiates the entire classroom and the lecture hall from just a random building with a bunch of students in it. The students are every other atom. They aren't as relevant when talking about organic molecules, but they are certainly a part. Organic compounds are essential for life. They're found in every living thing, hence the term organic. And as a result of that, the presence or creation of the first organic molecules on Earth was probably around the time that life started. A particular student who was interested in this was Stanley Miller. Miller, under the mentorship of Urey, who was his teacher, he performed a very simple experiment, trying to mimic the atmosphere of early Earth and see where these organic molecules may have come from. It starts with this apparatus. In this case, this water here was supposed to represent the sea. The water is then boiled, and the released water vapor, which is just the gaseous form of water, was then moved throughout this tube until it reached this compartment. This one was meant to represent the early atmosphere and contained 3 main gases: NH3, H2, hydrogen gas, and CH4, methane gas. You'll notice something interesting here. If we really are to get any sort of organic molecules out of this combination of water, ammonia, hydrogen gas, or methane, the only place that carbon can come from is this methane. There was also an electrode built into this, which basically sent sparks through the container. This would mimic the early atmosphere of the Earth, where there was a lot of lightning and storms, according to what scientists at the time had speculated. Now, this experiment took place in the 1950s, and since then, our knowledge of Earth's early atmosphere has changed, but the general ideas still remain the same. And this experiment has been repeated with other gases in the atmosphere and yielded very similar results. But what are those results? Well, first, as the vapor or the gas now starts to move downwards, it becomes condensed back into water. You'll have to pardon me for not drawing in the lines. I'm not very good at that. From this area, around the end, a small sample was taken periodically and kept aside for later study. This was repeated a lot. He took multiple samples as the water kept going back into this container to be boiled again. And he tested each sample for one thing: presence of organic compounds. Now, obviously, we wouldn't be talking about this if the tests weren't fruitful. So, yes, he did find organic compounds in most of these samples, which proved that his theory that the original Earth's atmosphere could have led to life through this method was confirmed, at least partially. Next, we have to talk about the unique bonding properties of carbon. To get there, we're going to have to take a step back and talk again about subatomic particles. You might know already that an atom can be drawn with this model, in which the nucleus is represented here with protons and neutrons in the center, and electrons are marked on outer shells, which can support increasing numbers of these electrons. The innermost shell can only support 2, and every shell afterwards supports 8. The outermost shell is called a valence shell. Carbon's valence shell in particular only has 4 electrons, and in covalent bonding, atoms are hungry to fill their own shells up, even in ionic bonding. Which means that carbon can still accept 4 more electrons that they're not able to get from anywhere other than outside atoms. Because of that, carbon can form up to 4 bonds. Recall that these bonds are just sharing of electrons, or even electron donation, depending on which bond we're talking about. Now, carbon can form a single bond, which means one bond to another molecule or atom. Can form double bonds, which is a pair of 2 bonds. Triple bonds between each other, and that's more exclusive to carbon. But at any point, it has to form 4 bonds. That's the normal property of carbon's bonding. This is another reason that carbon is one of the strongest elements. It's because it's able to form this many bonds and support this much tension. It also leads to some pretty interesting structures being formed. And obviously there are other atoms that can form 4 bonds, but none of them do it as relevantly as carbon. There's a few different shapes of carbon as well that we can talk about briefly. Carbon can form a straight chain like you've seen already. It can branch out in certain parts, like so. It can, for example, have varying positions for its double bonds. For example, this as opposed to this. And perhaps the most interesting one is that it can form rings. These are all very important structures that we'll talk about when the time comes, which is probably in the next video. For now, this is all we need to know about bonding properties of carbon. An isomer is any molecule that has the same chemical formula as anything else, but a different structural formula. A common example is C6H12O6. This is the formula for glucose, but also the formula for fructose. Meaning, they can have the exact same molecular formula, but their structures are different. This is a structural isomer. Another very common example is pentane, which is this straight chain of 5 carbons with hydrogen attached to each one, and so on. I'm just going to draw the lines here. This is pentane, and it has the formula C5H12. But another compound with the same formula of C5H12 is 2-methylbutane, which looks like this. It also has hydrogens attached to each carbon, but there's one key difference, and that's this branch. Because of this, you have the same number of hydrogens and the same number of carbons, but a completely different function. That's a structural isomer. We're also going to talk about cis-trans isomers. An example I want to look at here is this one. If you take a look at, say, these two hydrogens, if they're on the same side, this is called a cis isomer. But the thing is, when these bonds are doubled, they can't be rotated, which means I can't switch this chlorine and hydrogen around. If I were to switch the chlorine and hydrogen around on this side only and leave it the same on this side, you now have a trans isomer. Some of you may already know that trans means across, and it makes sense because the hydrogen atoms are now across from each other, as are the chlorine atoms. This is just one example, but this pair is called a cis-trans isomer pair. The last one I want to talk about are enantiomers. Enantiomers are a little bit harder to draw because there's not many common examples, but I'll just make something up for the sake of demonstration. If we look at, say, this molecule that I'm going to create on the spot. And you know what? Just to emphasize, I want to throw in, say, instead of a hydrogen, an oxygen. And on this side, the enantiomer of that would look like the mirror image. Very simply put, it's just like left and right hands, is an example that our textbook uses. If you look at your left hand with your thumb on this side and all your other fingers on this side, versus your right hand with all the other fingers first, and then your thumb. I'm talking about palm front, obviously. This is kind of what these look like. It's just one printed onto each other. And while it seems like this shouldn't have much of a difference, this can actually have a huge impact on their functions. For example, sometimes they even have opposite functions. If you look at molecules of ibuprofen, for example, the enantiomer of the effective ibuprofen molecule is a counter to the effective version. I realize now that I'm talking not about ibuprofen, but rather albuterol. But still, it can even decrease the efficiency or fully counteract the effective version. Or it can have a plethora of other effects. The idea here is that if you use the enantiomer of something by accident, it's not going to work well. That's it for this lesson on carbon, and while we're not done with Unit 4, we will have one more mini lesson to talk about one of the most important biological concepts of functional groups. This is important for biochemistry, physics, not so much physics, but it'll be very relevant in the future.